Progress, as we know, depends on more than just great research. It requires visionary leadership, strategic investment, and people willing to support bold ideas. Please join me in welcoming Dr. Arie Belldegrun, founder and senior managing partner of Vida Ventures, along with Dr. Michael Atkins and Dr. Tony Schwery, for a conversation on innovation, investment, and the future of kidney cancer research.
Three, yes. Okay. Arie, sit in the middle. Yeah. So I'm really delighted to have Arie Belldegrun for this fireside chat. And as an introduction, Arie was born in Tel Aviv and did his early medical training in Israel. Then he came to Harvard to do urology residency, and then went to the NCI surgery branch to work with Steve Rosenberg doing a surgical oncology and immunotherapy fellowship. And it's there that we first interacted. Now 4 decades ago. And following that, he went to UCLA, where he essentially built their urology program. He's currently the Roy and Carol Dumani Chair of Urologic Oncology, a research professor and chief of urologic oncology emeritus, and founder and director of the UCLA Institute of Urologic Oncology at the David Geffen School of Medicine at UCLA. But more than that, Arie, in addition to his academic career, has been a visionary entrepreneur. He founded Kite Pharma. I think I was in your home back in 2007 when you were just beginning Kite. It was at your dinner table that you were working on it. And that's a biopharmaceutical company that was engaged in innovative cancer therapies. And he was chairman and president and CEO until 2017, when it was acquired by Gilead. He's also worked in the development of several other companies, including Cougar Biotechnology, Agenesis, and Eurogen, and these companies have led to approval of drugs for prostate cancer, lymphoma, leukemia, and bladder cancer. And he's currently chairman of Belco Capital and founder and senior managing director of Vida Ventures, a life science venture group with offices in Boston, which brings him to Boston frequently, New York, and Los Angeles, and is also executive chairman and co-founder of Allogene Therapeutics, a clinical stage biotechnology company pioneering the development of allogeneic CAR T-cell products for cancer and autoimmune disease, including a trial that was just published in the Journal of Clinical Oncology, the Traverse trial in kidney cancer. We're delighted to have you here for this discussion, and I'm going to start with the first question for you, which is, tell us about your journey from urologic oncologist and researcher to medical entrepreneur, and what that experience has been like.
Well, thank you, Mike, for the kind introduction. It's great to be here in the Kidney Association, and this is actually my home, where I became a professor by writing all these papers in kidney cancer, and I moved from an assistant professor all the way to professor at UCLA. So I used to come every year and be here and be writing the grants, and I was with some of the spores, but you know, you move on, and in the last several years, I lost some track here. So I'm so delighted to sit here and to listen to really what's happening in the field. I trained here as a urologist in the surgery at Brigham and Women at the Harvard program in urology. I had a chairman who was a very inspiring guy, Ben Geddes, who told me, with your background, I did a PhD in immunology at the Weizmann, says, immunology and urology, you should go to see one of my ex-resident from the Brigham, Steve Rosenberg, and go to talk with him at the NIH. I think that's somebody that you need to combine with and do some work. I said, fine, I'll go to there. I've graduated, went to Steve, said, come to me, you'll be the first urologist I've ever trained. Well, if so, I came, and there was about 100 other medical oncologists, surgical oncologists working on melanoma, working on sarcoma, trying to see where will immunotherapy work. And being the best urologist in the group, I said, I'll work on kidney cancer. That's essentially how we started. And the idea was to check what is the immunotherapy that will best respond in any of them. So Suzanne Tupalian was the medical oncologist, surgical oncologist in melanoma, and made her name there. Jim Yang, actually still at the NIH, did sarcoma. Sarcoma didn't work, but he's moved on to other things. And I did kidney cancer, and suddenly kidney became an immunotherapy sensitive, not as much as melanoma, because we couldn't identify specific CTLs that recognize T cells exactly like in melanoma, but still enough to build my career in kidney cancer. And then I moved to UCLA. At UCLA, we started building a therapy, and as Mike pioneered the whole high-dose IL-2, I brought the high-dose IL-2 to UCLA, and it was a good anchor to build a kidney cancer program. We created a tumor bank of about 3,600 patients with 600 samples, nephrectomy samples that I took out from the operating rooms, normal and tumor, which essentially later on created a lot of value for us. And that's how essentially we developed the program at UCLA and moved on to be part of the kidney cancer community. In 1996, a group of 5-6 of us, professors at UCLA, said it's time to become a little bit more entrepreneurial. We are in California, we should be entrepreneurial. And we were sitting essentially together, said, what are we going to do? We'll take tumors we have in the bank, tumor bank, we'll take tumor, normal, and we'll try to identify what's the difference there. And we created a company called Agensis. Before Agensis, it was called Urogenesis. You can understand, because I was a urologist. So that was the idea. But then we decided, we identified tumors from breast cancer and others. So it's expressed in other tumors. So it's called Agensis. And Agensis, essentially, we took bladder cancer. We identified a new gene. We had no idea what it is. Owen Whitty was at that time the chair of the scientific advisory board with 5 members of the National Academy. And we said, Nectin-4. What is Nectin-4? Anybody knows. We try to understand what it is. We identify the nectin, and somebody said, create an antibody against it. And we said, great idea. So we made an antibody against Nectin-4. What do we do now? I'm going to the clinic. So actually, Peter Eisenman from Seattle said there's a new company in Seattle, just founded. It's called Seattle Genetic. And they are taking toxin and building it together with, bridging it together with a antibody. I said, okay, we have an antibody, let's do it. So we started, and we were the first one to actually create the antibody together with Nectin-4. And today, this is enfortumab, or Padcev. When you know how it happened and what happened behind that, of all the monkeys that died in the middle because the linkers didn't work, eventually it's Astellas, and actually I was yesterday morning at breakfast with the CEO of Astellas, and we were talking about 20 years ago of how we did it, and essentially that's how it started. So that gives us some flavor, let's do it again. So we went to Cougar, we developed Zytiga, we went to Mo, we went to Kite, we went to Allogene. You know, the appetite came with the food. That's it. So
Great.
I'm gonna, you know, Arie has been a friend for a long time. The first time I met him, Mike didn't mention that he's been on the first SPORE EAB, on the External Advisory Board. And I remember he's raising his hand. We're all talking about the effect of VEGF-TKI on the kidney, doing it neoadjuvant. And I don't know if you remember, and he's like,
sure,
look,
we talked about it.
We gave the patient, we gave this, and this is what you see. I'm like, wow, this guy is, so we started a friendship that is still lasting and hopefully for many, many years to come. One question, since really you covered my question in your answer, what is your view today about institution and academic researcher? How can they best address the federal funding environment for kidney cancer? There's a major gap. Do you think we should team up with pharma? In your opinion, what should we do if you had, you know, one thing to do? How can we address that? And I understand it's an open-ended question a bit.
It's an open, we can discuss it for a long time, but basically I've been discussing it with the Chancellor of UCLA, it's also Alan Garber a few months ago, and several times again, and they all agree. The question is how to implement it. They all agree that you cannot count for the future on the NIH grant or NCI grant or your SPORE grant, whatever it is. That's not the foundation that you have to build. You have to create an evergreen situation by which you can afford doing what you want to do, not because the constraint of financial. And that's about entrepreneurship. And that's exactly what we tried. I can tell you that we started, as I said, 5 of us. All of us are still professors at UCLA. We all continue to work in different companies. None of us left the academia. And I give it an example at UCLA with Owen Whitty. Yes, with all of the successes and everything, he still runs the lab, he does exactly what he has done 20 years ago, and Jim Economo and Tony Ribas. I mean, that's how we all operate. So that's not what's perceived as a conflict, I think, can be not a conflict. And entrepreneurship is something that that we take, and now essentially we have built in the last 6 years the California Institute of Immunology and Immunotherapy, which is a 750,000 square feet facility, which is hopefully will be open next year. We fought with the UC system to own 100% of the IP. No university owns any of the IP. If university gives something, they get in return part of the IP. And after 6 years of debates and lobbying and the governor in giving us the first $500 million to start the campaign, we essentially have now an institute that will be something unique. It will build what I call the Broad Institute of the West 21st century, with correcting everything that we learned from the Broad. And then there will be a whole section of startup companies where we will encourage anybody in the institute to develop his company there, and owns 100% of the IP. We'll decide what to do with that. The board of directors is a group of 2/3 of the board are entrepreneurs, Michael Milken, Gary Michelson. I mean, people that the community knows, and they are business people. And a third is the chancellor at UCLA, the dean of the medical school, but the majority is owned by business for business, and that's how it is. The third section will be a section that pharmaceutical will be homed there. So essentially, what we want to do is create a environment, essentially Candle Square in one place, in an organized and controlled fashion.
So I'll tell David McDermott, the future is bright, David, really bright. Should we open? I mean, we have a lot of questions.
I can tell you that Alan Garbus said, we should do it, you do it, entrepreneurial is the only way to do it in the future. So I said, okay, now let's see how we end.
The big question, it took you 6 years for UC to get the IP 100%.
It will take 300 years.
300 years from Harvard, and they will get 100% of the IP. That's the issue. I mean, if you can talk to Al, I mean, please.
You're 100% right. Yeah, you're right.
Mike, do you want to ask a question or open it? There's a lot of
I
people.
want to ask one more question.
Okay.
Please.
And then maybe follow up at the end with another question. But what efforts have you pursued in the kidney cancer space, and where do they stand? So this is your chance to talk about Allogene.
You know,
And others.
number one, for years in my research, we were interested in the HIF, and we took the carbonic anhydrase 9, the CA9, which we were lucky to bring from the Netherlands at that time, and it was probably 20 some years ago, bring the gene and do all of the research and published extensively on the CA9, try to popularize the work, or carbonic anhydrase 9 to do it as a imaging and or therapeutic or radiopharmaceutical, all of that section with CA9. I still believe that that's a very important molecule, despite the fact that we did what's the ERISER study, and it was 850 patients or so, and gave it as an adjuvant.
Rencarex.
The Rencarex, exactly. And it did not show significance, and therefore it's kind of the CA9 a little bit died. But I think that it's a highly expressed target on kidney cancer. How many do we have? And that's the problem. We have CA9. We are at Allogene now very excited about CD70. And CD70, we have created a CAR, an allogeneic off-the-shelf CAR, and it's, as you mentioned, it's this month in JCO. Unfortunately, the New England Journal of Medicine said it's this study is too small. I can tell you 51 patients. Out of that, we learned, obviously CD70 is expressed on, the pathologists will tell you 90-95% of kidney cancer express CD70. It's great. What we know today, it's not much different than what we learned later on from CA9. That's not so much what's expressed, but in what percentage, what's the density of the expression. What we found that out of 17 patients that had over 50% expression of CD70, we had 31% response in patients with kidney cancer. And I can tell you that I was impressed because, and Mike, you will know it, for years, we all treated patients with high dose interleukin-2 and other cytokines, but we have never seen anything happening inside the tumor. The kidney cancer, the primary tumor, had to be removed. That's how I did my thousands of nephrectomy. But we had to do immunotherapy plus nephrectomy. That's how it started. When we did here, this is off-the-shelf CAR T of CD70 CAR T that you give one single infusion, and then watch what happened in the kidney. The kidney shrunk significantly to almost, I'm not sure, we didn't do a biopsy, unfortunately, but I think even it might have been scar tissue. And when I saw that, after so many years of the primary tumor shrinking inside the tumor, it means that the CAR-T moved into the tumor, the TIL, the tumor-infiltrated lymphocyte, into the tumor, at that time overcame all the immunosuppression, the local immunosuppression, which is the major problem in all of these tumors, and the tumor shrunk. So clearly, and what we took in that study is patients who failed everything that you guys are treating here. Six different treatments, 5 different prior therapies. So I bet that if we take this now in an early stage disease, you will see significant difference. And I know that at the Dana-Farber, Wayne is doing now a CA9 CAR together with a CD70.
Correct.
I'd love to test it. Yeah, that's
So
great.
where does the Allogene,
We finished
product-
phase 1.
where are you going with it?
I'm applying to your grants, and we'll continue the phase 2.
Okay.
No. We definitely are very interested to continue that into a phase 2. It's essentially priority. We have now an MRD study with a minimal residual disease with Natera, and analyzing in patients who, after complete response to DLBCL of chemotherapy, and therefore that's our priority. It's hundreds of patients and multiple centers, but it's all priority.
Yeah, so hopefully you'll get to kidney cancer again.
Oh yeah, I mean that's.
So I'm going to open it up to the audience for any questions.
7 minutes for
Dr.
question. Dr. McKay.
Sure. I love what you all are building with the Immunology Institute. And it just got me thinking about what is the missing element. The missing element directly on that campus is direct access to patient care, patients getting treated on a clinical trial. Like, how can we co-locate a biotech hub where there's multiple, early biotech building drugs that are ready to get into first in human, and actually co-locate? I think that's lacking across the entire infrastructure. Like, I see there's I'm in San Diego, and there's so much potential to tear down the barriers and actually have true collaboration where we're bringing drugs directly into the clinic, getting answers quicker, testing faster. I think that's what it's going to take to, you heard what got released a month ago, kind of trailblazer from the Trump administration. This is how we're going to move the field forward,
In that particular case, I didn't mention that institute, UCLA is moving the Quantum Institute and the AI Institute to be in our building. So we'll have all of this, and the idea is just that you sit in the research, quantum, and AI together is significant. But next door, we are in discussion now to acquire another 300,000 square feet, which will bring it to over 1 million, in order to do phase 1 clinical trial, because all of that before is no patients. And that will be the idea to translate immediately from the drugs to, So the entrepreneurship is, what we do in academia, and I've done it for years, is we have a construct, we have something, we start talking with companies and sell our IP, maybe one patient or so. But think of that, if we can locally create a quick, not N of 1, but N of 10, and bring a company and show them in the university, look what we have performed here. That's what we have. This is the data. $100 million, not 500. That's part of the creating value. And that's when I'm saying entrepreneurship is creating value, and this will be an evergreen situation that you will not need your philanthropy, okay?
Well,
and
yeah.
that's the institute is in order to prevent philanthropy. Philanthropy will come now. We are raising, by the way, $1.5 billion additional. But once it's there and the institute will be inside, I hope the evergreen will create a situation, the IP there will not pay rent, pharma will pay rent, so that will generate enough research that we don't need philanthropy. My hope.
That's the best marketing strategy. Everybody gonna apply for this institute for a job.
Yeah, we are in discussion on the CEO right now.
Another
I think I
comment.
know.
How many hours do we have? I was very impressed in such a group, anywhere we go in the pharma, biotech, engineering community. It takes 5 minutes before somebody says China. Here it took a long time to say, but I can tell you that when I was at Kite, the CEO of Kite, we formed a partnership with Fosun, which is a big conglomerate in China. I was the co-chair and we're 50-50 partners there. And we brought to the FDA, to the Chinese FDA, an idea to develop CAR-T in China. They've never heard of a CAR-T, they didn't want to do CAR-T, but they said, you know what, we need to study it. Come back in next quarter, in 3 months, and we will see what we can do. We are coming, and the current CEO, actually, of Allogene, Zach Roberts, was at that time with me at Kite, and we went to China. They invited us, FDA, the Chinese FDA, invited us for 8-hour meeting. 8 hours. We are all dying to have a 60-minute meeting face-to-face now with our FDA. 8-hour meeting. We come in, there were 250 people. The question that we were asked were so sophisticated, we barely can ask. They did studies, and they learned what is CAR-T, what do we mean, and how, what reagents do we need, and whether they're approved already in China, the reagents, or they will have now a CAR-T that it's not approved because one of the reagents is not approved. That is, the sophistication. We built a manufacturing plant there. We got the approval of CAR-T, which today in CAR-T in China. And so we have the experience, and we have multiple companies that we are now doing doing clinical trials in China. I think that somebody needs to look and see why is it happening there and it cannot happen here, because it's not about innovation. But we have a lot of discussion. It's a very good question. We need to address it as soon as possible.
One more, maybe?
Are there other questions? Or Tony, do you want to ask the last question
And the last question, yeah, before
we break?
Well, we have one to think. So let's say money is not the issue, funding is not the issue. You know a lot about kidney cancer, and you're giving 5 years. You have 5 years to cure kidney cancer. What ideas, maybe 1 or 2, would you follow only?
You want me to fail to come in 5 years from now? I gave you all the money, I gave you everything you did, and you
5
didn't
years.
cure.
Luckily, I'm the one that has the money funding, and I'm giving it to you, and you have 5 years. Where will your bet be on?
Personally, with what I know right now, I would go with what we know about CD70 as a target and create the CAR, create an ADC, all the approaches that were discussed today, and go together and to analyze it. But you know, from what I've seen here, I mean, I don't have another target for anything in immunotherapy that I can come up with, which to me is right now is more exciting. And therefore, the combination of CNI and CD70 as a group and build around it a whole program, a project program, that's what I think. But again, that's one-sided view.
Well, you've had some good guesses in the past, and
Yep.
paid off, so maybe the same will happen here.
Thank you.
All right, thank you.
Stay here. We have